Our protocol utilized RNAseq and bioinformatical tools to quickly uncover the overexpressed driver genes that contribute to the formation of stem-like colorectal tumor spheres.
This protocol is used to screen the differentially expressed gene and the signal pathways to investigate the molecular mechanism for cancer stains in the selected colorectal cancers.
On day 7 of culture, use an inverted microscope with a digital cell imaging system to observe and measure the diameter of the tumor spheres. When the tumor spheres reach a greater than 100 micrometer diameter, treat the culture with 0.25% trypsin. After 5 minutes at 37 degrees Celsius, neutralize the trypsin with 2 times the volume of growth medium and count the cells using a hemocytometer. Collect the cells by centrifugation and resuspend the pellet at a 5 times 10 to the 4th cells per 100 microliters of medium concentration. Add 2 microliters of the primary antibody of interest to each tube and incubate the cells for 30 minutes at room temperature and 200 revolutions per minute. At the end of the incubation, wash each tube with 900 microliters of PBS and analyze the cells for expression of the protein of interest by flow cytometry, according to standard protocols. For RNA isolation, after trypsin collection.